And the purple looks kind of very nice.
Well, you're aware of it.
I don't have a purple shirt. Walmart and mine look really good. I mean, I have like 4 t-shirts, so like there's no way. There's no way. Yeah. I wear my baby shirt. Why should I have like this in what you for catching the shirt yourself? I know. I'm like oddly roundy up. I am like, is that your writing pen? That's what at least it prevents me from
Yeah, I know.
maybe, you never know.
All right, we'll get started. The very end of the last SI session, I was going over a problem like this. I went through it quickly, so I'll go over it again. Let me just write down some information on a piece of paper. Let's see. So, stranded. Okay, so, all right, so the question was simply, they give you the percentage base composition of single-stranded DNA. So the problem would tell you in a word problem format that in single-stranded DNA it was 40% G, 30% C, 20% T, and 10% A. And then the question instead now is asking what percentage of duplex DNA do you have either one of these? They won't ask for all 4, but they'll ask for either T, A, G, or C. So, a lot of people usually kind of slip up on this, but my way of solving it is if we know what one strand of DNA looks like, we know immediately what the other strand of DNA looks like. So, if this is 40% G, we can very quickly conclude that the other strand must be 40% C, correct? This one up here, if it's 30% C, that must mean that the one down here, yep, 30% G. Continuing along, that means this must be 20% A, and this must be 10% T. Okay, so we're almost at the answer there. If you add up all these percentages, it comes out to 200%, which is a little too much, but that's fine, because we can do a bit of a trick in a second. Let's say it was asking for what percentage of duplex DNA contains T. Well, let's see, that's that, and that's that. When we add that together, that becomes 30%, which is not the exact answer, because the very last thing we need to do is divide that by 2, and then we end up with our answer of 15%. So it turns out that duplex DNA is made of 15% T. Okay, yes?
Would you be able to get the same answer by just adding the A and the T percentages, just like alone, like the 20 and the 10 from the first one and dividing the 2?
This one and these two? Not necessarily, because if you're talking about like adding these two things together. No, because it may not always mathematically come out this way. Let me see.
Because I mean, the entire strand adds up to 100%.
Both of these together, yes, but that's not exactly right. Like, how to explain?
I mean, they give you like numbers that
basically, if you're adding just these two up, it doesn't necessarily tell you anything about the other strand exactly, but you can do whatever you want as long as it comes out to the right answer. I don't care. I'm just going with what method that I use personally.
Okay.
And once, of course, you have T, you don't even have to calculate everything else, right? If you know it's 15% T in duplex DNA, it's 15% A here. What is that, 30%, right? So that means these two, you have to divide 70% by 2, which is 35, 35. And a trick question, of course, is how much uracil is there in this? Very quick, very easily answer that it is 0%, right? Okay. Good. Not feeling good. All right. So back to this. Let's see. Oh yeah. No, that was a long time ago. But let's see if we can answer these things here. For each of the following, state whether it describes prokaryotic or eukaryotic mRNA. So we'll start with this one right here. Which one, whether prokaryotic or eukaryotic, are genes contiguous with each other? Prokaryotic. Prokaryotic genes are right next to each other. There's no gaps between genes, so they are contiguous, which is the same thing as continuous. So that is prokaryotic. Okay. Which of these organisms makes mRNA, where one mRNA can code for many proteins simultaneously? That is also prokaryotic. Good. Which ones have introns and exons in them? That is eukaryotic. Good. The phrase monocistronic mRNA is associated with which of these things here? That is eukaryotic, exactly. Okay. One mRNA can only code for one protein is eukaryotic. Now the tricky one here, one pre-mRNA codes for many proteins. That is eukaryotic, and honestly the key phrase is the phrase pre-mRNA because only eukaryotes make pre-mRNA.
Does pre-mRNA specify without introns then? Because with introns it doesn't code really anything, does it?
Well, it depends on definition of coding. I think coding is more general, like it contains enough of the gene pattern that eventually when you process it you can make multiple genes. Specifically, and let me see if there's a nice diagram in this one. Not really. It's in 10A, but there's a diagram, I think it's literally the last slide in 10A that shows that from the same pre-mRNA, depending on how you splice it one way or the other, you can make several proteins from that.
Right.
So that's the very last slide in 10A for you to see that. But yeah, that describes a eukaryotic. By the way, again, a synonym sometimes for pre-mRNA is heterogeneous mRNA, or actually heterogeneous RNA, which sometimes you'll see it written as HNRNA. Again, I kind of gave away the answer just now, but which one undergoes alternative splicing? Eukaryotic. And finally, this has to be prokaryotic, polycistronic, and the way I remember it, of course, is both start with the letter P, polycistronic prokaryotic. Okay. Polycistronic is basically the word that defines this phrase right here. Okay, so the one mRNA coding for many proteins, poly, is supposed to represent this many part. Okay, let's see. All right, yeah, you guys can probably easily answer this, but the process by which pre-mRNA is modified into final mRNA, what is that mechanism called? That is splicing, exactly. So, I'm just going to skip all that. Ribosome stuff, again, is easy. You could just look that stuff up, tRNA. Let me get to the siRNA. This is the slightly more complicated stuff. Let me put it on one page here. Oh yeah, I forgot about that. I haven't seen it in a while, but it does sometimes end up being a possible extra credit question, is the mechanism by which siRNA is made and what siRNA does. That used to be an extra credit question. I haven't seen it in a couple semesters though, but you have to know it anyway for your multiple choice questions at the minimum. Does siRNA prevent transcription or translation? Translation, because when does siRNA actually work, and we'll review it again in a second, siRNA only acts by binding to mRNA, meaning transcription has already happened. It can't do anything to transcription because it has already happened, yes?
I thought that miRNA was one that did translation.
Both act in the binding to mRNA. So siRNA does that, microRNA does that. There's another one called long non-coding RNA that does. Actually, long non-coding RNA can technically inhibit both, but that's a long story that you don't need to know. siRNA specifically only acts by binding to mRNA, meaning transcription has already happened. Therefore, the only technical step that is being prevented is translation of that mRNA. So siRNA is only able to prevent translation. Oh yeah, a neat little fact here. Knockdown or silencing of a gene expression by siRNA is permanent and the effect can transfer to daughter cells. So this is, if this was separated into two statements, it might be a little bit more clear. Since I didn't go over this, I'm going to talk about it right now. Let me show you. So it depends. It's a little bit weird. If we're talking about gene silencing in a single cell, siRNA is actually temporary. It lasts for about 7 to 10 days. Again, don't worry about memorizing the days part. It is temporary. But if within that span, that cell happens to divide, and you look at the daughter cells of that cell, those will actually retain that gene silencing thing permanently. So you won't be asked that vague question that I put on the worksheet. You'd be asked either one or the other. If they just say in general, is siRNA silencing a permanent thing? The answer is no. siRNA silencing only lasts for a couple days. But if it's asking, can the effect be carried down to the daughter cells? That answer is true if it was asking specifically that, is that gene silencing can be carried down to the daughter cells. So just stick with what is said on here. I made these problems like a long time ago, so I haven't edited them since then. Okay, so now we get to the mechanism. So like I said, you do need to know the mechanism for the exam, and at best, it could be your extra credit question. So arrange the following steps describing the synthesis and function of siRNA in the right order. We went over this last time. Does anyone remember of these 4 steps here? We'll just call this A, B, C, and D. Which one of these probably happens first? A, that is correct. Let's see what it says. Double-stranded RNA is cleaved by dicer into siRNA. So the first step talks about how siRNA is even made in the first place, so that is definitely step 1. All right, what happens next?
siRNA
Unwinds is the next step, exactly right. This is the next step. So if I go to a nice picture that illustrates this, the very first step when we talk about siRNA is listed here. Double-stranded RNA interacts with some enzyme called dicer, which explains what it does. It chops up the double-stranded RNA into siRNA. And then we move on to the next picture to see what happens next. The siRNA unwinds into single-stranded siRNA, which is the second step here. What's next? V, that is right. So, and then of course process of elimination, this last, D must be the last step. But let's see, step 3 is the RISC complex captures native mRNA that complements the siRNA. So that's this big picture here that you see here. Is that siRNA, that short fragment, will look for an mRNA that it is complementary to, and a bunch of enzymes put together called the RISC complex will bind with it. There are 2 possible results of this binding of the RISC complex to mRNA. The end result is still the same, no matter which pathway it goes down. But the end result is that no protein can be made because either 1, the mRNA was cut into pieces, therefore the ribosome can't translate the mRNA, or the RISC complex stays bound to the mRNA and the ribosome can't read the mRNA if there's this huge thing in the way. Okay. So end result, either way, is that it prevents translation of the mRNA. Yes, question?
Yeah, the previous question on the effect of RNAi being inherited.
Yes, that.
How?
How? That's something that's a little bit beyond what I know. For all I know, I think it has to do that there's some feedback mechanism with gene expression that probably gets carried down. I never learned that specifically, and it's probably beyond what you need to know for this class. I have no idea, but it is an interesting little fact there. Okay. All right. My guess is feedback, but I never learned exactly why, so I don't know. Yes?
Would it just be epigenetics at that point?
That's what I'm thinking, is that the fact that something isn't expressed highly maybe tricks the daughter cell into thinking that, well, maybe this isn't supposed to be made, and then there's some epigenetic pattern that carries that on. I have no idea. Okay. But good, you guys are thinking about it in a multitude of ways. That's actually very important. And if anyone's planning on eventually taking the MCAT, I can't tell you enough that the most important thing for the MCAT, by the way, is not like learning the material. It's thinking about things in a scientific mind. So this is good for testing, both in this class and in any standardized test. Okay, let's see. Blah, blah, blah. And again, some stuff you can look up, but yeah, what are some nice biomedical uses of interfering RNA? siRNA is just like the most common one, but technically microRNA is an interfering RNA. Long non-coding RNA is an interfering RNA. So the examples listed in your PowerPoints are a good start to kind of give you context on why this is important. All right, now we go back to the notes. I think that was the last thing we went over. Blah, blah, blah. Okay, let me zoom out real quick. So just for context purposes, microRNA is another type of, again, interfering RNA, and you actually see this made a lot during the developmental stages and when a tissue is differentiating. Again, more complex topics for a different class. So this is all this, you don't have to memorize which microRNAs are associated with which stages of development, just that development of an organism generally is associated with a large production of microRNAs, and then you're good. All right. Okay, differences between DNA and RNA. We actually talked about this a lot already in 10A, so just to review a little bit. I mentioned a long time ago that there's this weird situation between cytosine and uracil, which I don't think that, oh, yeah, there we go. There's a nice picture right here, is that spontaneously, just for whatever reason, cytosine will undergo a deamination hydrolysis reaction to become uracil. That's it. There's just some organic chemistry reason that we don't care about right now. Cytosine just transforms into uracil. That's a problem, right? Because DNA, you want to avoid mutations as much as possible, so there's a cool little trick that repair enzymes have figured out. Let's see. Yeah, so if you look at uracil here, and you compare it to thymine, you notice just one simple difference, and that there's a methyl group attached to carbon 5 in thymine, but uracil, there isn't anything there. This is a nice trait between uracil and thymine because it tells DNA repair enzymes traveling along your DNA as it's repairing and editing it, that if it finds uracil here, it's really just looking to see if there's a methyl group at that spot. If it doesn't find a methyl group at that spot, it knows that there's supposed to be cytosine there, so it removes the uracil and puts back cytosine because that's what was there before. If it just moves along and finds something that almost looks like uracil, but there is a methyl group there, it's going to leave it alone. That's why the specific phrase they use, that's why you get these weird phrases here. Natural uracil and mutant uracil. The mutant uracil describes uracil that is in DNA, which this obviously isn't supposed to be here. That is mutant uracil. It's not supposed to be there. Natural uracil, where are you supposed to find it? RNA. In RNA. Okay. Okay, let's see. More differences between DNA and RNA. Talking about the sugars, very easy. DNA is made of deoxyribose, RNA is made of regular ribose. Specifically, the difference is at carbon 2 in the sugar. They indicate carbons in sugars with the prime designation here, so 2'. Let's see if they have a nice picture. Sort of. Okay. So this here is an example of a ribonucleotide, or string of ribonucleotides. Here's the 2' OH here, and this page here actually explains why RNA is actually less stable than DNA. And I mentioned it briefly, is that when you have this free OH group, it likes to react with a bunch of different things. In the example they're giving here, it undergoes a reaction called base hydrolysis, where you see OH minus, which is a very basic anion, is going to react with this and cause this chain of reactions where it actually causes the polynucleotide to cleave itself. Okay, and that's simply because of the presence of this OH molecule. DNA does not have that there. It's just a hydrogen, which doesn't really react with anything, which explains why DNA is more stable. It does not cleave itself. So RNA, by its functional nature, is supposed to be temporary. RNA, as soon as it's transcribed, as soon as it's translated into a protein, we don't need it anymore. So it usually will degrade itself as time passes. So that's the difference between DNA and RNA. Other reactions that can be undergone by RNA and DNA are listed here. You do need to know these in case they show up as a multiple choice question or answer. So they're listed here for you. Again, the really important thing here is this. RNA can be hydrolyzed because of that OH group, and it does so by a mechanism called base hydrolysis. Okay. Okay, this stuff. So we'll go over this in detail. Let me find a nice example. Okay, let me write some stuff down real quick. Okay, we're going to use a 5' B cutter. Okay, so a common question that you see on the exam, and I've seen this on every exam 3 since I was SI and TA for Hasty, so you'll get something like this, where you'll be given a sequence, and it will have a phosphate written in somewhere. I forgot to add it on the worksheet. But you'll be given a sequence, and you'll be told some type of cutter is applied to this sequence, and you're supposed to look at the answer choices and pick which one represents the result of this cutter. So what these cutters are, you'll see in your notes, they say 5'A, 5'B, 3'A, 3'B. These are all exonucleases. And very important that you emphasize the exonuclease part, because there are similar enzymes called endonucleases that will do something different. So these are exonucleases. What these do is they will remove a nucleotide one at a time from an end of this sequence, from either the 5' end or the 3' end. These exonucleases will remove one nucleotide at a time. Using our context clues, probably the 5' B cutter is going to remove a nucleotide from the 5' end, which by now, hopefully you guys know that the left side of a nucleotide, that the left side of a nucleotide is the 5' side, and the right side of a nucleotide is a nucleotide sequence is a 3' side. So this 5' B cutter, by its nature, is supposed to try to remove this guanine off the 5' side here.
Ah, God. Wish I had tissues.
Ah.
Go to the bathroom today.
Yeah, if someone, you got something, that's fine. Thank you. Hopefully I'll feel better by next week. Okay. So that's the first part of the clue, is that we know that this cutter is going to remove guanine. The next thing is what they mean by B cutter, and that's what I'll try to illustrate here. So I'm going to change the drawing a little bit. Maybe we'll all be sick by next week. It's fine. Okay.
I don't want, I can't afford to get sick. I'm going on a boat.
Oh no, that would be the worst time.
It's for a different class. I'm going to be on it for 3 Great.
Okay, so what does this drawing mean? So this drawing is a shorthand notation for a polynucleotide, meaning the phosphates are just representing where those phosphodiester bonds are. Just for illustrative purposes, let me just go to some other nucleotide picture. Yeah, we'll just use this. So the ones on the left here, you'll notice that there's a nucleotide, a phosphate, another nucleotide, a phosphate, and so on and so on. So this is a shorthand notation for that. This represents the 5' end of the sequence, and this represents the 3' end of the sequence. So here's what an A cutter and a B cutter does. An A cutter will try to cut to the left side of a phosphate. That is what an A cutter does. You'll notice that I skipped this first phosphate. Why? Well, if I tried to cut to the left side of this first phosphate, have I really done anything? No, right? Remember way back with the amino acid sequences that if you try to cut a certain side, sometimes nothing happens. Well, these A and B cutters are going to cut at a particular spot where something will be removed. An A cutter will choose to do so by cutting before some phosphate. A B cutter will choose to cut something on the right side of some phosphate. Okay? And this could be applied to anywhere on here, but because these are exonucleases, we must do so at the very first or the very last nucleotide. And I'll go over a couple other examples in a bit. So, the 5' part of this name here tells us that G is going to be removed. The B part of this name tells us that it's going to cut after a particular phosphate. Therefore, the end result is we're going to get something like this. Okay. We are going to get a guanine that not only has a phosphate on the 5' end of it, it also has a phosphate on the 3' end of it. Well, why is that? Based off our picture here, the B cutter cut after this phosphate. So it's going to have to stay attached to the guanine because where else is it supposed to go? So this phosphate is going to stay attached to the guanine. The other fragment, which is the rest of the DNA sequence, remains as so, A, T, A, C, G, and there isn't anything here. In fact, what would be here, but not usually drawn, is a OH instead of a phosphate, because the phosphate that was supposed to be here is left with the guanine instead. Okay? So this is what a B cutter does. A feature of a B cutter is that it creates a fragment with 2 phosphates in it, and it creates a fragment with 2 OHs in it. That is what B cutters will do. Okay? For additional illustration, let's use the pictures for that. Zoom out. Is this the B? That's an A cutter. Here's a B cutter, I believe. They're cutting from the middle of the nucleotide, so that's not a good example. Here we go. Down here. Again, a B cutter, what it'll end up doing again, as you can see here, is it'll leave one fragment with 2 phosphates and another fragment, oh, they cut it all 5 times, excuse me. So, none of these are a good example to give you because they're a little bit misleading. But going back to this then.
So do A cutters basically make a fragment with 2 OHs in there?
Yeah, and well, no, A cutters I'll do in a second for illustrative purposes, but the pictures on there make it look like the exonucleases will do all of these simultaneously, so I'll emphasize then with these pictures. Exonucleases can only do these cuts one at a time, and they can only cut one nucleotide at a time from the ends of this DNA sequence. And you'd only be asked which of the following answer choices represents the fragments you would get from either an A cutter or a B cutter. So this would be your answer for this particular question here, is that you would get a fragment that is written as PGP, and you'd get this other fragment here. It'll most likely in answer choice format be written as ATACG, because for whatever reason people don't like to draw in the OHs. So these are the 2 fragments you wo' get if a 5' B cutter was used. Okay. I'll do a couple other examples with the other cutters so that you can compare and contrast these things to each other. Okay. All right, what was the G-A-T-A-C-G? Okay, so this was the original thing. Let's do 3' A cutter this time. All right, the 3' part tells me that this is going to be the nucleotide that needs to be removed because it's at the very end. The A part tells me exactly how. So let me draw that nice shortcut diagram. Five, 6. Okay. So the 3' part tells me this has to be removed somehow. The A cutter part tells me it'll do so by cutting on the left side of a particular phosphate, as indicated right there. This is the A cutter. Therefore, what we are left with is, and I like to draw on the OHs, but the answer choices might not do that. Okay, so we are left with these two fragments if a 3' A cutter was used. A feature of A cutters is they will always produce a fragment with a 5' phosphate and a fragment with a 3' OH. Both of these will look like that. Okay? And if you were to do another round of a 3' A cutter, it will move to the next nucleotide and begin doing the exact same thing. It's going to try to remove cytosine. It will do so by cutting to the left of a phosphate right there, and it will make an additional fragment and do it over and over and over again. So, from these, there are 4 possible cutters you can get. Right? 5' A cutter, 3' A cutter, 5' B cutter, and 3' B cutter. We've gone over 3' A and 5' B, but there's obviously a couple other possibilities here. I'll leave those to you guys. But you guys have the tools to solve this now. Again, the trick is the number tells you which end of the DNA sequence will have its nucleotide removed. The letter will tell you how it will be removed, either cutting to the left of a phosphate or to the right of a phosphate. Okay. And that's my way of solving these things. Sometimes other people have a slightly different explanation. You'll get the same answer either way. So do whatever makes you feel most comfortable. All right. So let me make sure that's good for the exonucleotides, exonucleases. Let's see. Yeah. All right. So any questions on exonucleases? Okay. All right. Next topic. Bacteriophages, of which you do not need to know all these cycles of stuff and everything. Probably in a microbiome course you need to know this, but you really don't need to know this for this course. So moving along. The really cool part, talking about bacteriophages, but more general, are these enzymes called restriction enzymes. Restriction enzymes go by another name, and I don't remember if it's on this slide or another slide, called endonucleases. So we learned about exonucleases, whose job is to remove nucleotides one at a time from the ends. Restriction enzymes are also called endonucleases, E-N-D-O. It's weird that they don't have it on here, but if you see the phrase endonuclease, it's the same thing as a restriction enzyme. So what these things do is they look for a particular pattern. Man, does he not have a single example? All right, this is the closest example. I'll use this to describe what a restriction enzyme does. A restriction enzyme, what it does, EcoR1 is an example of one, is it cuts somewhere in the middle of a DNA sequence. If an exonuclease cuts on the outside, one nucleotide at a time, an endonuclease jumps straight in the middle, looking for a particular sequence to cut. Endonucleases, or restriction enzymes, because they're synonyms of each other, they do this by looking for a particular pattern in DNA. When it finds that pattern, every time it finds that pattern, it will perform 2 cuts. one on each strand of DNA. So this example, the restriction enzyme EcoR1, you don't have to memorize it, but what it was doing is it was looking for the pattern GAATTC, and when it does so, it cuts right here between this G and this A, and it performs a second cut right there between that G and that A as well. It does, and again, by cutting in the middle of a DNA sequence, you open it up. I'll get to more of this detail later. I don't know if I'll be able to do it today. But that has enormous application, and a lot of interesting science happens simply because of restriction enzymes having the ability to cut an area of DNA. So I'll probably talk about it in a little bit. But in terms of properties and everything, because it is able to cut DNA, it does so in two potential ways, and I do not like that this is the only example. The one way is it creates what's called sticky ends or staggered ends. What that means is, and EcoR1 is a good example of this, when it cuts at these two places, it creates non-overlapping regions of DNA, indicated in pink here. Those are called sticky ends or staggered ends. They're kind of like a step ladder here. Because of this, because you have these non-overlapping regions here, you can fuse together different DNA sequences into your newly opened strand of DNA. So when a restriction enzyme cuts DNA into this staggered pattern like this, you have these small regions of non-overlapping DNA which you can replace with something else. You can do gene editing because of this small little detail here. Gene editing, which is again editing a gene sequence by cutting it open and putting something else in. All right. So that is one application of restriction enzymes. The other potential pattern the other potential form it can cut is, other than sticky/staggered ends, is blunt ends. Blunt ends, there's really no application for. Blunt ends, they're just creates, I guess this would be the best example here, is you have two flat ends like this. Because of these two flat ends, you can't really fit anything together, so we don't talk about blunt cuts really at all. Again, this section of the notes, there's a lot of detail, but you only really need to know certain specific things which are covered in the worksheets. Blah, blah, blah. This is some history of restriction enzymes. The very first one is EcoR1, whatever. Okay, this is a lot of math to do with restriction enzyme cutting. I'm going to skip it for now because I've rarely seen this on the exam. If we need it for the exam, I'll go over it, but it takes a long time to explain when it might not even show up on the exam. So I will go over this during the review if it turns out this is going to be on the exam, so I'll skip that. This is most likely going to be on the exam, the information here, so I'm going to cover it right now. Let me just draw something. Okay. So, let me just draw some stuff. 1, 2, 3. This one is okay. All right. So applications of restriction enzymes and endonucleases. There's a technique called restriction fragment length polymorphism. It's a technique, but it also describes a property of genes, of a genome. So all this really means is that all along your genome, you will have DNA patterns that restriction enzymes like to cut. All right, the example earlier was EcoR1 likes to look for GAATCG or something like that. If it finds that pattern, it will cut it no matter how many times that pattern exists in your genome. So what this ends up doing is that if you're doing gene editing or you're analyzing some genome, you take a DNA sample, you apply a particular restriction enzyme, and it's going to cut it into different sized pieces because these DNA patterns are going to show up at different places in your genome. In this example here, they talk about one gene that comes in two forms. If you've taken genetics, you learn about that there's like a dominant phenotype versus a recessive phenotype or anything like that. That's not really covered in detail in this course, so all I'm going to say is both of these represent the same gene. This one is just the dominant one that shows up in genetics in this example, and this one is the recessive one. In genetics, we use capital letters to indicate the dominant one, the more common one, and lowercase letters to indicate the recessive one. Bottom line, because these are the same gene but a slight difference between them, that slight difference makes it so that some restriction enzyme, I'll call it restriction enzyme X, we still got time, right? Yeah, we got a couple minutes. That restriction enzyme X is going to cut at the labeled spots here on this gene. But on the recessive version, because there's a small difference in it, this restriction enzyme is only going to cut at these two label spots. So notice that for this one up here, we create a small fragment and a medium-sized fragment if this restriction enzyme X was used on the dominant gene. If restriction enzyme X was used on the recessive gene, we create a really long fragment of DNA instead. This difference can be visualized. Oh yeah, let me just draw that line in. This difference can be visualized on a gel. Remember those gel electrophoresis stuff that we saw in exam 1. And you might remember that near the bottom of a gel, you have shorter things show up here. And near the top of the gel, you have longer things, longer fragments show up at the top of the gel, right? So if I were to draw a couple lines there, this one up here would represent the longest fragment. This one down here would represent the next longest fragment, the medium or the short fragment here. So keep that in mind, because then there's one additional detail here, is this line here, okay? So if this was not here, if these lines were not here, we wouldn't be able to visualize any of these fragments. What these lines represent is that when you actually do this experiment, you attach a fluorescent label to your DNA. You know, wherever it goes, it's going to stay there, and you can visualize any DNA fragments that come about. So right here, there's a fluorescent label that will go to this fragment on the dominant gene, and a fluorescent label that would go to this fragment on the recessive gene. Therefore, when you look at a gel, the line right here, which represents the longer fragment, must represent lowercase a gene, okay? The line down here, which represents the shorter fragment, we see on our diagram here, must represent the capital A gene. This technique, and I'll review it again next SI session, just in case, because it's really important, allows you to take a bunch of different DNA samples, and depending on which lines show up and which lines don't, you can figure out whether they have the dominant or recessive particular gene. Okay? So, if someone had a genotype where they only had the dominant gene, they will probably only have a line down here. If someone only had the recessive gene, they should only have one line showing up at the top here. And if someone is heterozygous, meaning they have a copy of each of these, they should show two lines. Does that make sense? I know we went over it a bit quick, and I'll go over it again next time because it looks like we have to leave because there's a class in here. But the bottom line is this gel here you can read and you can figure out what genotype a particular patient has. I have seen this type of question on every semester that I've been working here, so we'll go over it in a little bit more detail next time. All right, that's it for now. If you haven't signed in, the sign-in sheet is going to be up here. And